So thank you for all coming, and now I'm going to hand it over to Lee.
All right, welcome everyone. My name is Lee Frame, and I'm the Program Director for Integrative Medicine here. We have a master's degree program in integrative medicine, and it's one of the leaders in the field, and I would argue probably the most evidence-based. So if you know anyone who's looking to get a good grounding in integrative medicine, please send them our way. But without further ado, I'd like to introduce Dr. Emeran Mayer. He received his combined MD-PhD degree from the Ludwig Maximilian University in Munich, Germany. He completed his residency at the Vancouver General Hospital in Vancouver, Canada, and his gastrointestinal fellowship training at the University of California at Los Angeles, UCLA, as we must know it. Dr. Mayer has a career-long interest in clinical research aspects of brain-body interactions, with a long-standing focus on the bidirectional communication between the brain and the gut in health and disease. He's recognized as one of the leading investigators in the world of brain-gut interactions in GI disorders, including functional and inflammatory bowel disorders and obesity, with continual NIH funding since 1989, which is pretty impressive. Dr. Mayer is a professor of physiology and psychiatry in the Division of Digestive Diseases at the Geffen School of Medicine at UCLA. He's also the executive director of the G. Oppenheimer Center for Neurobiology of Stress and Resilience and co-director of CURE, Digestive Diseases Research Center at UCLA. And he's also the best-selling author of The Mind-Gut Connection, How the Hidden Conversation Within Our Bodies Impacts Our Mood and Our Choices and Our Overall Health. Please welcome Dr. Emeran Mayer.
Thank you, Lee, for the kind introduction. It's a pleasure to be here and an honor. I'd like to thank the Song family as well and John Penn. So we realize that our connection goes back a couple of decades when we were both part of the early phases of the collaborative centers of integrative medicine, academic centers of integrative medicine, a long time ago. Just for myself, I have a career-long interest in brain-body interactions. That's why I went into medical school. I'm a relative, considering my length of career, relative newcomer to the microbiome field, and we got into this because it became logical that if you're interested in brain-gut interactions, that you have to include the microbiome. That dawned on us about 7 or 8 years ago, and since then, so really been passionate about this topic. I'm going to give you sort of a tour de force through an area that ranges really from the gut, from the brain, the gut, to the planet, to the soil. And it will be fast, a lot of slides, but I did want to do this because I've become really passionate about looking at this connection, not just the gut and the brain, but also how this relates to the larger environment in which we live in. So I have some potential conflict of interest, but don't receive money from these entities. It's not stopped working with the pharmaceutical industry some 10 years ago, and none of the things that I'm showing was really related to these commercial entities. One thing that became pretty clear to us that we had to amend the concept that we had of brain-gut interactions. So 10 years ago, this looked a lot simpler. So you had a brain and a gut, and there was a circular connection. So in the meantime, we know there's multiple loops involved in this, including in the body, the brain immune loop, the microbiome immune loop, direct loop between the microbiome and the brain. But one thing became very important is that we're also very closely connected with a extensive loop with the environment, particularly, as you'll see at the end, the soil microbiome. The connections with the environment are both in terms of psychosocial factors, but also importantly in terms of diet. So it's really a holistic view. I'm sure 10 years from now, this will further expand and get more complicated as we learn more about this, but I think it's a framework that I'll base my presentation on. So rather than give you the summary at the end, I'll start out with the summary at the beginning. So after that, you could basically turn off your attention. So the key points that I want to make is that communication between the gut microbiome and the brain is an ubiquitous, ancient, evolutionary conserved system that modulates host behavior. This communication connects the mind, the gut, food, and the environment in a way that can best be understood in a systems biological way, not in a reductionistic linear way. Key components of plant-based diets act through the gut microbiome to produce gut and brain health. And the soil microbiome has a major influence on the health of the plants, the gut microbiome, and the brain. If you get deeper into this, you realize that many of our problems of health go all the way back to what we're doing with the soil and the food that the way we grow the food in this current industrial environment. Let me start out with the brain-gut microbiome axis. So there's a ubiquitous, this is not something new that started 10 years ago when the first papers came out about, hard to believe, these early papers, that somehow the gut microbiome played a role in modulating or influencing the behavior of laboratory mice. In the meantime, we realize there's a lot of evidence that has been accumulating even before that. So now we know the affective, social, pain-related, and digestive behavior of mice is in some ways influenced by the gut microbiome. You know that mating preferences, locomotion, aging in insects, social interaction and communication in hyenas. Then we also know that there's a relationship between the gut microbiome and molecular processes in the brain. And we'll talk about this a lot more. There's a lot of things, the last composite of this, which has been found really by comparing mice that have been grown up in a germ-free environment, so-called germ-free or neurobiologic mice, and mice with a normal means normal for the laboratory environment, which is definitely not normal for wild mice. But anyway, so we know that if you take away the microbes in the gut, there's a lot of developmental changes that happen in most of the brain function and developmental function. As humans, we have co-evolved with trillions of these microorganisms that inhabit the human body. The problem, they are invisible, so we haven't really paid much attention. What we don't see or feel directly, we have not paid attention. So this went unnoticed until not that long ago, and now obviously there's a lot of enthusiasm, a lot of data that we have microbial ecosystems that are distinct in each of those locations, different parts of the body. One of the earliest ones was the stomach, so H. pylori, that organism that was detected and then it's been implicated as a major risk factor for peptic ulcer disease, not the cause, but a major risk factor. But now we know there's also a microbiome in the mouth plays a major role in tooth decay and gum disease. Even around the belly button, a unique distribution of composition of microbiomes. Every part of the body or the skin has a unique set of these microbial communities, which strangely don't mix. So every time you take a shower, you would think they were all mixed up. They're so uniquely adapted to their unique environment. And the same thing probably happens inside of us. So those are in the small intestine, the stomach, the large intestine, have all their own properties and stay where they are. In the intestine, this is clearly the area of great interest here. And in the large intestine, the largest concentration of these microbes, hundreds of trillions, probably in the universe in one place. So why would there any connection between the microbes in our gut and brain functions? I always compare this. So the dirtiest organ in the body and the cleanest, most sophisticated supercomputer in our brain. Well, you have to go back in evolution and you realize that for over 3.5 billion years, microbes have inhabited, were the only inhabitants of the oceans of the world, had a lot of time to develop their communication skills, develop the language. Some 500 million years ago, they settled one of these microorganisms, an algae, settled in a unique animal, one of the first marine animals, this what's called a hydra, basically a floating digestive tube with a nerve net around it. And some of these algae settled inside of that floating tube, got free transportation, free and constant delivery of nutrients, and was protected. So it turned out to be very advantageous. And because of this design principle has persisted throughout evolution, virtually every animal that exists today, from flies to rats to hyenas to elephants and humans, have maintained that symbiotic relationship with the microbial world. What we had known for some time, that is shown on the left side, what microbes do is they break down molecules that we don't have the machinery in the small intestine to break down and absorb. These elements of food were very prominent early in our evolution. So our ancestors and even our primate cousins had mainly a plant-based diet, had even more of these. So the specialization was to break down these molecules, and different ones, fiber, plant-based fiber, resistant starch, one of the major ones, turn them into short-chain fatty acids and then could be absorbed in the colon. Substances called lignans, which would turn into phytoestrogens. Polyphenols turn into secondary metabolites. And also what we call xenobiotics, so molecules that these microbes have never seen in their billions of years of evolution. They're able to break them down into metabolites of these. And sometimes we're not even aware what those metabolites of drugs, of pesticides, then do to our health. On the right side is, so these breakdown products are not, they don't stay in the gut. It's been shown that they function as signaling molecules to most of our other organ systems, including the cardiovascular system, the immune system, enteric nervous system, but also brain and behavior. Just to get a sort of an idea of the magnitude of this system is that, so we all know we have about 20,000 human genes, but we've realized that we have up to 20 million microbial genes. The function of many of these genes is still not known, so there's a big question mark and what's coming up in the future. What's to me most interesting, that we could write our textbooks and practice medicine by ignoring 99% of the total gene pool of what's been called a holobiome, basically the microbes and the humans together. This is obviously a huge opportunity for future development and treatment and diagnostic possibilities, that we have a lot of unknown substances that are potentially of benefit for health. This concept that these breakdown products of microbes that produce these signaling molecules, it's very complicated. It's not just that we have tons of genes, millions of genes. We also have, it's been estimated, hundreds of thousands of these metabolites. And they don't act by themselves, but there's networks of these metabolites, so the metabolome. Anytime we deal with multiple elements, we give it an -ome ending. And it's really the end result of these interactions with these metabolites that have then the effect on human health. A very daunting task for the pharmaceutical industry, obviously. So on one side, a huge opportunity. On the other hand, a daunting task to figure out. This is not the traditional pharmaceutical model where you identify one molecule, one receptor. You have to really look at what these interactions of these molecules do ultimately to human health and what role they play in disease. Let me give you a couple of examples how the microbes can talk to our systems in our body. So the communication with the immune system is obvious. There have to be a close communication because these trillions of microbes are only separated by microns from the gut-based immune system. The majority of our immune system is located in the gut, so-called gut-associated immune system. And what you see here on this on the schematic is You have the lumen of the gut, you have a mucus layer which separates the microbes from it's called the epithelial layer or the lining of the gut. And then you have the immune system right underneath that epithelial layer. It's been shown that certain microbes, beneficial microbes, can they produce these metabolites like the short-chain fatty acids, butyrate or others, that can interact with the epithelial system and affect certain immune molecules in a different ways during development. Early on, in training the immune system to recognize things that are bad and good for us, but also later to have a highly functioning immune system that does not result in inflammation. So if you imagine this design, an amazing engineering design, you put potentially harmful, trillions of potentially harmful molecules or organisms right next to the immune system that could cause havoc within the body, that evolution was able to separate those two, even though they're so close together, by the gut barrier, the mucus layer, and the epithelial layer, which normally under intact conditions are very tight and will not let anything go through. On the right side, you can see what can happen in situations of inflammation. It's an example from inflammatory bowel disease. The point that I want to emphasize is the immune cells that receive these signals from the microbes, not all of them stay in the gut. They actually, so here are some of these molecules that make it to the immune system. So they activate dendritic cells, a subtype of immune cells. These dendritic cells produce cytokines, inflammatory molecules. These cytokines, some of them can go through the blood-brain barrier, either directly or through a mechanism that sort of jumps across this blood-brain barrier, get in contact with microglial cells. More than half the cells in the brain are not nerve cells, but these immune-like cells, which again produce cytokines, which affect nerve cells, neurons. And this is this phenomenon that many people have learned when they have, or tend to assume when they have conditions like brain fog or lack of concentration, so chronic fatigue. It's thought that some kind of a neuroinflammation plays a role. We also think that if this situation persists long enough, that you can get neurodegeneration as we have it associated with cognitive decline. So a very well-studied connection between the microbes in the gut barrier, the immune system, and then what happens upstream towards systems within the brain. Another communication channel, so imagine the microbes in the brain interact via different communication channels. So this is this neuroendocrine channel. We also know quite a bit about this. So in green, you see a specialized cell that's embedded in this epithelial layer in the gut. These cells are called enterochromaffin cells. And so in the meantime, everybody knows about this, but a few years ago, we had this talk in psychiatry. Psychiatrists was out, how can so much serotonin be in the gut? 99% of the serotonin in our body is stored and synthesized in these cells. This enzyme TPH is the rate-limiting enzyme that produces serotonin from tryptophan. And here comes the amazing story. The microbes, these spore-forming Clostridiales, have been shown to produce metabolites such as these short-chain fatty acids from plant-based foods and secondary bile acids that stimulate this enzyme to produce serotonin. It's been estimated that 60% of that stimulus, that serotonin production, is influenced by the microbes. And what's equally interesting is that so the microbes can't produce serotonin themselves, so they stimulate these cells. These cells release serotonin back into the gut, and then the serotonin molecules in the gut interact with the microbes. It's recently, Elaine Zhao, collaborator at UCLA, it's recently shown, it's not published yet, that some of these microbes contain the serotonin reuptake mechanisms, serotonin transporter. So they actually transport the serotonin into the cells. So when you take an SSRI, an antidepressant, it doesn't just affect the brain, it affects these enterochromatin cells and the microbes as well. What effect this has overall on our on this communication system, we don't know yet. If you eat, another thing that's important now, if you eat a lot of tryptophan-containing foods, it will stimulate these microbes to produce more of the serotonin. That's also really intriguing because serotonin, these cells are connected via synapses to vagal afferent nerves that carry that signal, the serotonin signal, directly into brain limbic and autonomic centers. So this gives you a good illustration of how food, the microbes, the gut, specialized cells in the gut, and the brain interact, and almost certainly play some role in the regulation of our mood states and emotional states. Exactly what role that is, we know this in mice, but we can't really say this in humans yet. Similar situation for another type of cell, the endocrine cells. I said earlier, the biggest part of the immune system is in the gut. It's also the biggest part of the endocrine system is in our gut, distributed in all these different cell types. So the satiety hormones that are released when you eat a meal, and these meal components interact with these cells, release these satiety hormones, which are supposed to shut off your appetite and your craving for food. Very similar design so we always thought it's the food that directly interacts with these cells, but now we know that microbes also through this mechanism of short-chain fatty acids have a major effect on the release of these satiety molecules, which then go both in the systemic circulation, reach the brain, but they also go through that same mechanism of an endocrine neural connection into the nucleus tractus solitarius. And so this seems to be a general design principle. And yes?
So what's the SCFA stand for?
Short-chain fatty acids. So I started in the beginning saying that the main breakdown product of plant fiber are short-chain fatty acids. Certainly not the only one. There's 400,000 metabolites. But one that's particularly well studied are these molecules. And so what I'm telling you here is a very reductionistic view. Just imagine that tens of thousands of metabolites produced by the microbes interact with these cells that determine our well-being, our mood state, and our appetite. Don't look at any details of this. I just want to say something. Microbes, based all on animal studies, play a major role in brain development. I'll come back to this concept again. The first 3 years of life is a period where your gut microbiome is programmed with a lot of interactions with the developing brain. So many essential brain development steps, so the blood-brain barrier, development of brain's neurogenesis, the microglia regulation, myelination of parts of the nervous system, and neural survival, growth, and signaling have all been shown to be influenced by some of these microbial metabolites. So the brain that we have the development is to a big degree influenced what happens early on in life, and that's why these germ-free animals that grow up without any microbes have major defects in any of these neuronal developmental phases. And that's summarized here. So during a lifetime, there's different stages where diseases develop and where the microbiome has been implicated. So developmental disorders, many diseases of the brain are considered developmental disorders. They don't just start when you're 40 or 50, but they have a long trajectory. And the role that interactions of the microbiome with the brain, this interaction has most likely a major influence during this phase. If I had to pick one period in lifetime where I think that it has to get the most bang for the buck if you intervene or understand this, is during this 3-year developmental period. And it's just like a software, like an operating system. Once that operating system is laid down, it will stay with you for the rest of your life. You can put on different programs, but the operating system will essentially stay the same. It's kind of ironic that we don't really pay that much attention to this. If you think about all the things that can go wrong and influence this developmental phase, from disease of the mother, nutrition of the mother, stress that the mother is experienced during pregnancy, all of these have been shown in animal models have an effect on the development of the infant's microbiome. Obviously, the antibiotic intake, which is vast. I forgot if I brought that slide with me, but by the age of 3 years, there's I think 2.5 doses of antibiotics that kids get, even today with all the data we know about this. And then there's clearly after delivery, the mode of delivery and the early diet, breastfeeding, which is influenced by the diet of the mother. So we haven't really paid that much attention to it. I think this is one area that will need a lot of attention for nutritionists, for OBGYN physicians, to really minimize the damage that we do during this crucial operating phase. So here are the numbers. It's staggering, particularly the last one, almost 3 courses by the age of 2, 11 courses by the age of 10. Every time, if you take as an adult an antibiotic, it has a transient effect, and it will come back, most people, to the pre-antibiotic stage. If you do this in an infant with a developing gut microbiome, it will decrease and probably even lead to the extinction of certain species of microbes. One way that I like to sort of demonstrate this, what happens early in life. So from a systems biology or ecosystem viewpoint, so this is a landscape of stable states and unstable states within the microbiome. The deeper the holes are, the more stable that state is. That state is determined by the microbial diversity and abundance. So the more diverse, the more stable that system is, the deeper the stability holes are. So early life experiences and genetics are the main factors that influence the creation of that stability landscape. Now, what happens later, particularly in North America, Western world in general, we are in a pretty unhealthy diet overall. So the microbiome and this complicated system is exposed to often a lifelong exposure of things, and we'll come back to this, that will have an influence on not only the operating system, but on the relative functioning of our microbiome, with influences on other functions in the body. Metabolic syndrome, the epidemic that we're currently going through, is a good example, and the microbiome has been involved in this. And then one area that's received a lot of attention recently in terms of funding is also the possible role of a lifelong exposure to an unhealthy environment for the microbes in terms of increasing the risk for neurodegenerative disorders, Parkinson's, Alzheimer's, and cognitive decline. There are big NIH studies, multi-center NIH studies going on to evaluate this role. So coming back to the stability landscape, in bolded, you see the factors that as an adult, what the system that was programmed early in life is exposed to. So perturbations that can be that can be stressful life events, diet, this is on the left side, mind-brain factors, perturbations also, chemicals, and exposure to potentially harmful substances, and lifestyle. So we know that exercise and social connectedness, probably through signals that the brain sends down towards the system, can also influence this. So summary that we can't change the operating system, but we can modulate whatever we got early on in life in significant ways by having a healthy lifestyle that includes diet, but also other factors. So the model that we've been working with, it's a systems view of these brain-gut microbiome interactions. You have a circular interactions. The brain can influence the gut in a major way. The gut can interact with the microbes in the bidirectional way. And the microbes can feed back to the brain with their metabolites, but the brain can also exert a major influence on the microbes directly. And I'll show you a couple of examples. So what are the predictions from a systems view of that? So this is better understood really if you talk to an engineer. The engineers are really good in talking about system science and what the properties are. So once established, the system is stable and relatively resilient to perturbations. Everybody in this room has been exposed to antibiotics as an adult. You probably had a relatively minor effect on your system of this microbial system. You can go fasting for a week, starving the microbes. When you eat again, they will come back the way they were before. Even people that undergo this C. difficile colitis that wipes out most of the microbes, in some miraculous way, it can be reconstituted from some memory that's in our gut. So it's a very resistant and resilient systems. This also implies that most therapeutic interventions in the adult have small and transient effects on the system behavior. Mentioned the antibiotics, also fecal microbial transplant. Even though it works well once you knock out the normal system in C. difficile colitis, it's not working well if you have an intact ecosystem in the gut, and efforts have been made for in IBS, in IBD, in obesity, and in autism spectrum disorders, and it doesn't work well. It's kind of amazing, but explainable by the stability of the system. Another thing, most individual acute perturbations in the adult have relatively little effects on the system output. Just showing a few examples again. It doesn't matter when you're stressed, even though during stress you change your microbiome composition and metabolites. When you get back into a relaxed state, it will bounce back to that same system. And then there's another property that any system that has these feedback loops, so one from the brain and then to the gut and from the gut to the brain, exhibits oscillations. It's not a stable, static, it's not a static system. And there are variations diurnally, food-related, and seasonal, and I'll show you one example for this. This has become very clinically relevant because, as probably some of you know, intermittent fasting has sort of become different forms of that. It's become a popular concept that people that do it think it's the best thing if you want to lose weight and maintain your lower weight. What this shows you, this paper from the Weizmann Institute in Tel Aviv. So you have, this is again the gut cross-section, the microbes that are in the lumen of the gut, the ones that are adherent to the epithelial layer and can directly communicate. These are the different phases: the dark phase, the light phase, dark phase, which was kept constant in these animals, what's called regular in these animals. So what you can see is during the dark phase, you have almost the disappearance of these. Actually, 12. You almost have a disappearance of these microbes that are close to the epithelial layer. During the eating phase, you see the microbes coming back, and this repeats itself. So during the dark phase, you have this increase in these mucosally adherent microbes, and then they disappear again, almost like completely wiped away from the surface of the gut. So when we I mean, the main thing about this rhythm is that mice obviously don't eat when it's light, so they have the reverse cycle. But as humans, when we sleep, we get into a situation that is shown here, where these microbes pretty much disappear and don't continue to signal, and they come back in the morning. So what is happening with this time-restricted eating or intermittent fasting, that you want the system to oscillate between this state and this state. Most of our modern lifestyle is, so you eat much longer. You have kind of wiped out this distinct separation between fasting, physiological fasting, and eating. You come home, you have dinner, you sit in front of the TV, you eat snacks, you have wine, and so you give this phase a much shorter time than it would normally be if you had a 12-hour sleeping rhythm. So what these diet approaches do is they try to bring it back to you have a 16-hour fasting period, which basically means you don't eat anything after 7, you skip breakfast, but everything else can stay the same. And you will shift the system back into its normal oscillatory state. It's also been shown, at least in mouse experiments, that a disruption of the circadian rhythm plays a big role in the development of obesity and metabolic syndrome. So people that are particularly affected, that do a lot of traveling between time zones and totally mess up their normal rhythm, this has been shown there's an increased risk of metabolic syndrome. So some of us who do that, unfortunately, professionally, to travel between time zones constantly, are exposed to it, as well as shift workers like nurses who are also challenge this physiological rhythm. And this is important because if you look at this now, this is the nighttime, this is the daytime. You see that you have different differences in these what's called mucosal associated bacteria. They can interact closely with the microbiome. This affects gene expression profiles in the gut. So this is not just happening with the microbes. They signal to the gut cells, change their gene expression profiles. The signals go into the circulation and reach other organs like the liver and the brain. So this circadian cycling and the shifting between feeding and eating has major effects on our homeostatic system. It's only sort of been recently this has come into focus, both therapeutically but also from a research standpoint. So like many ecosystems, the more regular it oscillates, the healthier it is for the system. So what happens during stress? I told you acute stress has a very small effect on this complex system. But what happens under chronic stress? So if you look at this concept, so the stress can be directed at the brain, disease-related, environmental, or it can be directed at the gut, infections, diet, antibiotics, toxins. And we can sort of conceptualize this and see what is the output of the overall system. So when you have a stress on one part of it, on the brain, it doesn't stay in the brain. It will affect all the other components that are in this complex system. The same, the gut. If you perturb the gut, it will influence the brain and behavioral functions. We should give a couple of examples about stress. So stress does a lot of things to the home, to the environment in which the microbes live. I won't get into the details of this, but in decades of research, all these have been shown to be modulated by stress. An important one, for example, regional motility or transit times in the gut will change the habitat of the microbes, and if it increases the motility and decreases the transit time, you will have fewer microbes in that environment. But there's many other, including the immune system. Another study that showed a pretty substantial effect of stress, again in a mouse model, on both the gut microbial composition, but also on the downstream effect of what the microbes produce. So in this model of chronic variable stress in mice, they found that this had a major effect on the microbial abundance with a decrease in lactobacilli. So lactobacilli play a major role in tryptophan metabolism. So this molecule we talked about earlier plays a big role in serotonin synthesis. Lactobacilli produce a substance, hydrogen peroxide, that inhibits an enzyme that turns tryptophan into another substance with an unpronounceable name, kynurenine, that has been shown to have many negative effects on the nervous system, including neurodegeneration, neuroinflammation. And in this particular model, they also showed that the behavior of these mice, called the depression-like behavior, increased in parallel with a decrease in the lactobacilli. So the stress acted on the behavior indirectly through influencing what the microbes produce. In this mouse model, unfortunately, we can't do this in humans. This has always been the big hope of people that want to sell probiotics. It could be completely reversed by giving back the missing lactobacillus. So in these mice, when they're stressed, if you give them the lactobacillus as a supplement, it will reverse these changes. Unfortunately, this is not so easy in humans. Another aspect of stress is really intriguing, that stress also can have a direct effect on the microbes themselves. So one is on the environment and the habitat. The other one is directly on the microbes. And so you all know that when we stress, our heart rate goes up, the blood pressure goes up due to release of norepinephrine. Now we also know that norepinephrine doesn't stay in our body, but it leaks into the lumen of the gut and interacts with what's called adrenergic-like receptors on microbes, and it changes the behavior of these microbes. So when you're stressed, your microbes behave differently. So they don't just change in their distribution and relative abundance, they also behave differently. This has been shown for many pathogens, for example, that they become more aggressive and virulent in attacking the host. So when you get a gastroenteritis, you better don't be stressed, because there's a likelihood that it's going to last 2 or 3 times as long and you have much more severe symptoms. And that's related to this mechanism. It's been studied mainly in farm animals when they're taken to the slaughterhouse, where these enteric infections are very common. So for the sake of time, I think I'm just going to leave it at that and skip over a series of slides that go through all these individuals, what we know so far. But because of a lot of animal data and a few slowly growing data in humans, changes in the microbiome, a so-called dysbiotic state, have been implicated in a variety of brain disorders, from depression, autism spectrum, Parkinson's, multiple sclerosis. Just a couple of words to this. There's growing evidence from humans now that in depression, the microbes are actually involved. And you can see this on this previous mouse study. We may know the mechanisms by which this happens. Autism spectrum disorder, definitely an alteration in the microbiome composition that has been demonstrated and in mouse models can be reproduced. There's even an open label study with fecal microbial transplant in kids with autism that surprisingly had a follow-up study just last week, where they found that after several months of this open label intervention, the kids were still less symptomatic that received it. So that's ground for optimism. Parkinson's disease, in multiple sclerosis, it's a different mechanism. I told you this early programming of the operating system, interaction with the immune system plays a major role. This, I think, is pretty much established also in humans. So the increase in various autoimmune diseases, including multiple sclerosis and food allergies, is thought to be related to this faulty programming of the immune system, and the interactions with the microbes are implicated and play a significant role. So I'm going to skip over a series of slides here. I'm sure you would be interested to hear about this. So there's a lot of examples from humans now. The summary of that is that these microbial changes don't cause these diseases. They're modulatory factors. If you start out with looking at healthy individuals, asymptomatic, with genetic susceptibility or vulnerability to one or more what we call polygenic disorders that require multiple genes, most of these people stay healthy, but if something happens to their microbiome, the interaction of this genetic vulnerability with the dysbiotic state is likely to enhance the risk of these people then developing an actual disease. What percentage that is, we don't really know, but it's certainly not a one-to-one. So if your microbiome is compromised, there's no guarantee or no need to worry that you're going to develop Alzheimer's disease. It's one modulatory factor. And the factors in the environment that play a role in this are these triggering factors: chronic infections, chemicals, so pesticides, diet, and stress. So we can't change this part, but we clearly have ways of influencing these environmental trigger factors. So let me use the last few minutes, and I'll skip over some of the slides about diet and the microbiome system. So this is, in the meantime, fairly well known to a lot of people from the lay press. So if somebody is on a healthy diet with a high nutrient to calorie ratio, which is typically a diverse plant-based diet, high in fiber, polyphenols, polyunsaturated fatty acids, we have an intact mucus layer, and I told you this before that this is part of the barrier function. We have an intact, tight epithelial layer. We have immune cells that produce anti-inflammatory cytokines like IL-10, and we have a highly diverse microbiome with high abundance of certain microbes that, for example, play a big role in production of the mucus layer, like this Akkermansia, eosinophilia. So what happens if we switch, and probably, I don't know the percentage in this room, probably 30-40% would be in this category. So on a diet with a low nutrient and high caloric ratio, so these are high fat diets, low in fiber, high in refined carbohydrates. Several things happen. So the abundance of the microbes decreases, particularly of some of these organisms that produce the mucus. The microbes get closer, because the mucus layer shrinks, get closer to all these sensors in the epithelial layer, and there's probably hundreds for microbial products. This will lead ultimately to a decrease in the tightness of these junctions, and overall the combination of decreased mucus layer thickness and quality and increased permeability of the epithelial cells leads to a compromised gut barrier with activation of the immune system in the gut, which then distributes systemically and affects other systems in the body, probably all systems from the cardiovascular system to adipose tissue, but also the brain. And this process, this chronic immune activation, starting in the gut, to a large degree influenced by our diet, leads to these to a state that now a lot of people think, and in science everything that seems simple is usually not true at the end, that this chronic immune activation is sort of the answer to a lot of our chronic diseases. It may be true, but being a scientific skeptic, I think time will confirm that. Showed you this slide earlier without the diet. So this complex system is directly influenced by what we feed our microbes. We currently, if you take a single supplement with a single substance, like here, it's at a high concentration, or the same with a probiotic, it's most likely not going to have the same effect as this, system that has evolved over hundreds of thousands of years. It's probably one of the most intelligent systems in our body. And we know, so again, I'm going to skip over this, we know largely plant-based diets have the greatest health benefit. So I hope there's nobody in here that is a proponent of a ketogenic or paleo diet. Always surprises me that there's still people that propose that as the optimal diet in view of the overwhelming evidence. Many studies, large-scale studies that have shown the same, came to the same conclusion, not necessarily specific to the Mediterranean diet, but to any diet that is largely plant-based. There's also studies that this affects directly the brain, so not just the overall health, but has direct structural effects on the brain in older subjects. One example from studies that we know, such a diet will affect the glial cells, these immune-like cells in the brain, and change their called the phenotypic state of these cells, being incompetent or being incompetent, producing a lot of cytokines, again playing a role in this neuroinflammation. So there's many mechanisms that have been implicated. So the high ratio of plant to animal-based foods, same for fats, high percentage of omega-3, short-chain fatty acids, high concentration of polyphenols from olive oil, red wine, nuts, and fruits. And again, the microbiome comes in here because it plays a major role as an interface between these dietary benefits and what happens to us. Health-wise, the breakdown products of many of the foods, the plants that we eat, including these healthy polyphenols, or also called antioxidants, a minimal amount gets absorbed in our small intestine. The majority is broken down by the microbes and produce these health benefits. I showed a couple more, so I'm a few minutes over, a couple more slides on this. So I think when we think how important the plant-based food is for microbial health in many aspects, and how the microbial health plays a key role in transmitting widespread effects on our body, including the brain, it's obviously important where these plants come from and what they have been exposed to. Not every plant that looks the same contains the same amount of nutrients. So I'm thinking particularly about these polyphenols. A plant that's grown with chemical fertilizers does not have the same amount of polyphenol concentrations. It may look beautiful, twice as big as, so for example, blueberries. You can buy these gigantic blueberries that may be completely depleted in these health beneficial molecules, because what they produce is an interaction that they have with the soil. Shown here, so I highlighted the involvement of polyphenols in the interaction of a plant through its root system with the microbes in the soil. And this is an amazing concept, that the interaction of the microbes with the root system plays a big role in plant health. So when the plant is sick, sends a signal to the roots then communicate that to the microbes. The microbes produce these same polyphenol molecules that the plant transports up into its leaves and fruits and berries as the plant's own medicine. So when we, and chemical fertilization interferes with that process in the soil. So when we eat something that's grown in a regenerative organic soil without chemical fertilizers, it will have a lot more of these molecules in its berries and nuts. Good example. I once was in Italy. A friend of mine, actually, whom I'm going to visit tomorrow. He harvests olives from 800-year-old trees that he sort of acquired because people are not really paying attention to those trees. When he harvests the olives, these are all healthy trees. When he harvests the olives, he measures the polyphenol content right after harvest, and it's several-fold higher from any other olive oil that you could buy. So a good example that how the plant, these trees have probably survived and stayed healthy because they have produced a optimal amount of this medicine, this plant medicine themselves, and we can transfer it to our human benefit. So I'm going to end with this. So there has been a connection, so closing the circle really. I don't know if anybody in this audience has read this Lancet EAT report, where it's been clearly indicated this integrated relationship of planetary health with individual human health. In short, the conclusion is in order to make food production sustainable into the next decades, it has to shift towards a plant-based diet. Otherwise, the effects on climate change would be unavoidable. So transformation in healthy diets by 2050 will require substantial dietary shifts. Global consumption of fruits, vegetables, nuts, and legumes will have to double, and consumption of foods such as red meat, sugar will have to be reduced by more than 50%. Conclusion, a diet in plant-based foods and with few animal-sourced foods confers both improved health and environmental benefits. So this report, I would highly recommend everybody to read, has been attacked, to my surprise, by a lot of people on social media, but also by aspects of the food industry. And it's been criticized because they say it's been subsidized by Monsanto and all kinds of evil companies, which is actually not true if you go back to the sources of this. A fairly independent commission of very well-respected scientists, including Walter Willett from Harvard Public Health School. I think this is a beautiful example that we now have rationales for medical reasons, for gut health, for brain health reasons, but also now for planetary reasons, that the shift towards such a diet is absolutely unquestionable and will have to happen if you want to stay healthy and avoid catastrophic climate change. So my last slide, I've started as a reductionistic trained gastroenterologist who didn't have not even an hour of education in nutrition, which I still find amazing, still the same thing today. And really through this microbiome science, and I would say microbiome science is, if you had to pick one reason why it's so important, is because it has shifted the mindset of people involved in it towards this ecological, holistic worldview, which takes you all the way from the molecular basis to planetary health. And I hope it's become like a mission of mine to convey this message. I don't do it at UCLA because UCLA is too molecular and too reductionistic. Nobody's interested in that really. But I think talking to audiences, international audiences, I think people are really receptive, particularly younger people are very receptive to this concept. So thanks for your attention. Sorry I've gone over time.
All right, we're going to do questions now. If someone would help me with some microphones by passing out, Misha, can you help me? Questions?
Hi, are there any commercially available gut mapping options at this point?
Well, there's several, and on my disclosure slide, I showed that I'm on the scientific advisory board of several of those. So I have a direct insight in what that field is. I don't want to say anything negative about this. I think it's a very exciting approach. Some of these companies are further along than others. A company called Day 2, which was founded by the people that did these diurnal variation studies from the Weizmann Institute, which basically testing will give you an indication if you have metabolic syndrome and diabetes, what diet is optimal for you, and they base it essentially on the microbiome assessment. That will be available by prescription very soon. So they've provided probably the best science of all of these. The others are on their way. The principle this is based on, that they collect hundreds of thousands of samples from different patients and then apply artificial intelligence supercomputers to extract information that is custom tailored to particular subgroups of individuals. So I think it's not quite there yet, but I would bet my money on it, or if I had sufficient money to do this, invest in these companies, because I think this will be an essential part of medical assessment that you don't have to see a doctor for. The problem right now is there's so many out there that offer these services. I don't see a patient in my clinic who doesn't come with one of these reports, and I would say they're not scientifically supported, or the conclusions that they make from these is scientifically supported. I think ultimately it's big data science, and for that it takes a while before you get all these numbers together. But I think it's going to be a great way. You don't even have to go to the doctor. You can do this at home. So that the concept is you would order this online and do it at home.
Hi, thank you so much for coming to speak with us. You mentioned the role of antibiotics in affecting the long-term gut health of young children or babies, or whether they're fetuses. I wanted to ask if you know of any current research that is seeing any type of intervention that can restore the microbe population, especially for patients who absolutely need these antibiotics when they're young.
Yeah, so the current thought is that in the West, we have decreased our diversity of the microbial ecosystem by about 40%. So this goes along with the same number that we've done to our natural environment, external environment. And the thought is that within a certain bandwidth, you can compensate for that by healthy diet, plant-based diet. But you cannot bring back the organisms that have gone extinct, just like extinct species in the external environment. There's a lot of efforts, and this extinction probably happens fairly early, because they have compared the children of Yanomami Indians on the Orinoco River, infants with North American infants, and even in the infants in North America, there's already this 40% decrease in the diversity. So the big challenge, and people are working on this, is there any way, like fecal microbial transplant, ingestion of microorganisms or consortia of microorganisms that would bring back these extinct species? And one of the most interesting approaches is that you would have to do this early on in life. And not from the mother, because the mother already has this decreased diversity. So you'd have to do something, a synthetic mix of microorganisms that is able to reconstitute during development this diversity. And there are efforts to actually do this. It will take a while. It's risky if you do something to a 3-year-old infant. What could be the long-term consequences? But if you listen to Martin Glaser, who's written the book The Missing Microbe, when he gives his talks, he is extremely concerned about if we don't find a way to change that, where this will take us. Look at any other ecosystem in the natural world. If the diversity goes down to a certain level, there's catastrophic events, the stability disappears. And that could happen to our microbial ecosystem as well as to our external. So the interesting thing to me is that this is happening in parallel inside of us as it happens outside of us, in the soil and with animals, diversity around us. So right now, not a good way. I think still eating healthy is the only way you can do, but you will stay in a certain bandwidth. You'll not come up to the optimal diversity.
Yeah, and follow up to that, I'm concerned that we are turning this whole thing into another intervention. Okay, how to treat this. So we need to get to the place where we don't need to really treat this. And maybe we can let the kids run out in the yards and not tell them not to eat the dirt and drink the rainwater.
But it's actually beyond the dirt. I think, so we now know that nutrition of pregnant mothers plays a big role in, and that could be really the missing link. these Yanomami women have this high diversity and their infants have it, because of what they eat. I mean, they have a largely plant-based diet. Even though they're surrounded by hundreds, thousands of different animals, they don't overeat. They eat their manioko and their roots and tubers. So it's quite possible that interventions in healthy pregnant mothers could do a big part of this job, which would be the way, probably should be looking at. Yes.
Thank you. Based on your information, you said those bacteria are very important for our GI environmental. But what do you think about the chemo treatment? And also, those are very toxic. And also, for example, Lyme disease, people usually treat for more than a month with antibiotic. So in that case, are we in the wrong direction treating patients?
Yeah, so I've seen patients with Lyme disease who have been on 5 years of broad-spectrum antibiotics. At some point, we're thinking about doing a study and analyzing their gut microbiome. It must be greatly degraded. And what role that plays in the chronicity disease, we don't really know, because so it's quite possible this is a vicious cycle. They start out with one problem, then you decimate their gut microbiome. That feeds back to the brain and makes it a more chronic and resistant problem. It's just my guess. I'm not an expert on Lyme disease.
How about the temperature?
Yeah, this is a whole other story, an exciting story that has kind of emerged recently, that the microbial composition relative abundances appears to be a predictor of some forms of cancer therapy. And it's pretty clear that chemotherapy will probably wipe out, it's not been studied, will wipe out a lot of these microbial species. But it's really the immunotherapies that appear to be predicted by the baseline state. So I think people have started in oncology to look at that as a factor that could predict the outcome. So that's an area that doesn't involve the brain necessarily, but that may have sort of major implications to do a baseline testing of your microbial profile. I should also say one quick thing. So a lot of the science is based on the traditional, relatively old-fashioned way of studying microbes. It's the sequencing, so you just get relative abundances of microbes. You don't get absolute counts. You don't get the genetic content. You don't get the metabolites that they produce. This is rapidly shifting now to these other, more accurate techniques. So I think a lot of the concepts that we have right now, based on this early phase of microbiome analysis, will probably have to be modified. I think many things that you've read so far may not hold up to these more elaborate and sensitive techniques of analyzing.
I'm wondering if you can comment on the role of the vaginal microbiome in any of these changes that we see with the diminished biodiversity. Is that something that has been studied? Is it connected to the gut microbiome in some way?
I'm not sure if I really understood that.
The vaginal microbiome, like, is that?
The vaginal microbiome, yeah. So that's a totally different ecosystem, much less diverse in terms of species, but highly diverse in terms of strains. So it's a different design principle. And so the vaginal microbiome is affected by stress of the mother, for example, to a significant degree. And in mice, it's not been reproduced in humans. If mice are mildly stressed, pregnant mice are mildly stressed during pregnancy, changes the microbiome. When the babies are born, they get inoculated by a very different microbiome than from a non-stressed mother. And that will affect brain development in these mice, where only the mother has been stressed, and the transmitting mechanism is the vaginal microbiome. The vaginal microbiome also may play an important role in other, like in human diseases. vulvodynia, interstitial cystitis that affect the pelvic organs. And that's about all. I mean, we've been involved in these kind of studies. That's all I can say. I think the most intriguing thing is that this could be a mechanism that translates what's happening to the mother to the infant by when the infant is born and goes through the vaginal canal, is exposed to a different initial set of microbes.
Padham's Chaudhry did this interconnected series. Were you part of the interconnected series videos that were done? Was Padham Chaudhry did this series?
Yeah, so I was interviewed for that. I sort of have a little bit of mixed feelings.
That was my question, was what were your feelings?
And I did it against the advice of administrative people at UCLA who thought that this was just an infomercial, that entire film, which it turned out to be, unfortunately. So it's, yeah, I mean, I didn't say anything. They didn't leave that much in that I said because I didn't agree with the commercial aspects. So, yeah, so that's...
Okay.
So you have to be careful. Some of these documentaries that are coming out now about, the gut microbiome and the mind-gut connections are heavily influenced by commercial interests and really geared towards selling products that fit into that narrative.
Okay, thank you.
Hi. My question is, what is your opinion of the low FODMAP diet? And how is that diet affecting the microbiome? And do you think it's a good idea that people with IBS and potentially other brain-gut disorders are trying that diet?
Yeah, so I do not recommend this. It's the worst thing you can do for your gut microbiome. It works temporarily because it starves the microbes so they produce less gas. The problem in IBS is really the hypersensitivity that IBS patients have in the gut to many stimuli, including distension and gas. And when you look at it, so I emphasize this concept that your gut microbiome is programmed early in life. So if you go through this for a few weeks while you do it, yes, it will decrease the diversity and the gas-producing bacteria. But the minute you stop it, it will come back. So, and it's pretty clear now from comparative studies, you don't have to do that and then reintroduce certain elements later. You can actually do with an elimination diet where you use the patient's own insight and experience. If they don't tolerate something in a consistent way, some vegetable or something that produces more gas than others, they can decrease the intake of that particular item or eliminate it. It's much better, I think, to involve the patient in the management rather than kind of going to give you this standardized diet. And again, it's the word, you cannot stay on that diet because all the things that I said about what's healthy for the microbiome and the gut and the brain, you're doing the opposite. So yeah, I don't use it in my practice.
Okay, that's all the time we have. I'm sorry, I know we could ask you probably 5 million more questions. Thank you very much.
Okay, you're welcome.